From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 4, recorded on January 6th, 2020. I'm Vincent Racaniello, and you are listening to the podcast all about the nervous system. Joining me here today in New York City, Andres Bendesky. Welcome back.
Hi, nice to be here.
Missed you last time.
Yes, I missed it last time. Sorry.
And also right here at Columbia, New York City, Jason Shepard. Welcome.
Thanks. It's great to be here.
In vivo.
Yeah, in vivo, in the flesh.
So you're visiting Columbia, right? You're giving a talk tomorrow.
Giving a talk tomorrow.
And you came in yesterday, right?
Last night, yep.
And in your honor, we gave you a little dusting of snow.
Yeah. It was pretty cold last night.
It was our first snow of the season, I think.
Oh, really?
It was less than an inch, though.
Yeah.
Did you see anything outside in the
Not
cars? Really. No, not really.
You woke up too late. I saw it at 8 a.m.
Okay.
Yeah, in the suburbs, which is where I am, there is some very little dusting. It's probably going to be gone by the time I get back. But you can even see it across the way there, the Hudson River. Is your first visit to Columbia?
It is actually, yeah. I've never been to Columbia.
You've been to New York, I presume.
Yeah, many times to New York.
All right. Well, I hope you enjoy it.
Will do.
In honor of your visit, we're going to talk about your work. And it's just the three of us because Ori and
Aaron.
they're not available. Ori's back on service, which means, he's an MD PhD, and after he finished his PhD, now he's got to go finish learning to be a doctor. And so he couldn't make it today. Today's his first day on service, so hopefully in the future we'll be able to work around his schedule. If you like what we do on this and all the other related podcasts of Microbe TV, consider supporting us financially. You can go to microbe.tv/contribute. We have different ways you can do that. We'll come back to that later. So last time we talked about addiction. Now we're going to talk about Jason's addiction.
Yeah, you could say it's addiction.
Science is an addiction, right? So you a couple of years ago published this lovely paper in Cell, which we covered on TWiV. And the name of that episode was Raiders of the Lost Ark.
Nice.
Isn't that great?
I love it.
I told you that before.
I love it, yes.
I'll tell you that again. But there's nothing like getting it from the horse's mouth. So let's talk about it again.
Yeah.
This was a Cell paper, right?
It was. Well, yeah, stepping back, what I'm really interested in is how memory works, and fascinated by the fact that almost all the proteins in our brain turn over within minutes to hours, but we can, remember things for up to a lifetime. So that question of
some things, right?
Yeah, some things. That question of how you get the persistence of memory is really actually the main question that my lab is interested in. We've taken a very simplistic approach, and so I guess my addiction is this one protein called ARC, and I actually started working on ARC even as a undergrad. Well, when I was an undergrad in New Zealand, I wrote an essay on ARC, and I thought, this is a really interesting protein. Let's figure out what it does. And I'm still working on it.
So how long has it been known as a protein?
So two labs actually cloned it independently in 1995.
Mm-hmm.
And what they were doing they were looking for genes that would be induced by learning. So we know that when you learn, there's this whole gene program that gets turned on. And one of the genes that's most dynamic is ARC. And so in a brain from an animal or us that's just sitting in sitting in a familiar environment, there's not a lot of expression. But as soon as you put someone in or an animal in a learning environment or a new environment, then you get induction, the transcription of this gene immediately.
So what did they clone it from? What kind of organism?
So from actually from rats, I think. And at that time, it was somewhat crude. So what the way they would do this is they knew that if you have lots of activity in the brain, you can induce these genes. So they'd actually induce a seizure, an artificial seizure using electric convulsive shock. And that's how they had this sort of big signal to noise of their screen. So back in those days, they were doing these differential screens to look for genes.
Right. Nowadays, you'd probably do RNA-seq.
Yeah, exactly.
Look for differences, right?
Yeah.
So when you walked into this room the first time, you're making more ARC, right?
Yeah, hopefully.
Because it's a new environment.
Exactly. So we think that ARC is really tightly coupled to experience, and so much so that actually most neuroscientists are familiar with the gene because many groups have used it as a marker for which neurons are active during a particular behavior. And so it's one of these immediate early genes. Another famous immediate early gene is FOS, CFOS.
So what does ARC stand for?
So ARC actually originally stands for, Paul Worley's group up at Hopkins cloned it, and they called it activity regulated cytoskeletal protein. Partly because it was, at the time, they thought it co-sedimented with actin and may be involved in actin modification. The other name you may see is ARG 3.1, so ARG, and that was from Dietmar Kuhl's lab in Germany.
Okay. So there's a word for the animals that produce it. What is it? I forgot. It's in your paper. Everything with legs.
Tetrapods?
Tetrapods, yes.
Okay.
Four legs.
Right? Trust the evolutionary biologist to get what you're saying here. No,
all tetrapods produce ARC, right?
Yeah. So the evolutionary story here, I think, is actually really fascinating, and we weren't really aware of it until we stumbled on this viral homology. So yeah, it seems that the gene is in all land animals, all tetrapods, but it's not in fish or any lower vertebrates. But then there's a fly gene that looks like ARC, and we can get to that. But that actually, we think that that's another independently derived gene. So both of these genes seem to have come from the retrotransposon family, the gypsy retrotransposon family.
Right.
And so these are viral-like elements that can jump around in genomes. But I think what's fascinating here is that here you've got an actual gene that's been re-derived from one of these retrotransposon sequences, and it's maintained a lot of the ancestral function, and we'll get into that.
So is this produced in multiple tissues or just neuronal tissues?
So we think it's mostly in brain, but within brain it's only in neurons. It's only transcribed in neurons. There's potentially some peripheral tissues. There's one kind of immune cell, dendritic immune cell, that seems to express it as well, but it's fairly restricted. So we think most of its activity is in the brain.
How many genes come from retrotransposons or look like gag in tetrapods or mammals, or do you know?
Yeah, so we're interested in this. There was a couple of papers. One paper showed there's at least 30 families of genes in the human genome that have gag-like homology, but I think that's probably I think once we get into delving into this sort of tertiary structure, that we'll figure there are more of these. Even the retroviruses, if you compare across families, if you just look at amino acid conservation, it's not a lot, but it's really in the structure. So I think there's going to be a few.
So I noticed in this paper you use knockout mice for ARC. I guess those were made a while before you published this. Probably when the gene was first cloned, right? Yeah,
so we've had the knockout for a while, and those mice, if you look at them, they run around, they seem normal, so the brain is wide up properly. But what is interesting is that if you look at memory in these mice, they can learn. So if you sort of immediately look at their ability to remember right after the learning task, they're fine. But come back a day later and there's just no recollection. So there's no storage or consolidation of memory. So we think that what ARC is important for is some sort of molecular mechanism that converts short-term into long-term memory.
So such mice don't have any other issues as far as you can tell.
As far as we can tell, I mean, we're still, I think, delving into their behavior, and we have new mice that we're able to do conditional knockouts, which will, we worry that there's some compensation for in the full germline knockouts, but the fact that there's this really severe defect in consolidation, but not a lot of other things, is actually good for us because it gives us a real handle, I think, on this idea of persistence of memory. And so that's why I've sort of latched onto this one gene, is that if you're interested in consolidation of memory, it's, we think, necessary. And so if we can figure out why, it'll give us a clue, I think, on how all of this works.
So the gene was cloned in the 90s, you say, and people must have known shortly after that this homology with retrotransposon protein.
No.
They didn't.
Yeah, so this is where, science is, we're in our silos. So when I started working on this in Paul Wally's group as a graduate student, so I think I started in 2003, certainly we didn't know anything about that. And I actually remember running, in those days, an ancient blast search.
Yeah.
And gag-pol came up. I actually remember this. But then I was like, oh, this is, what is this? Why would I even pay attention to that? And so I forgot about it for forever. And it's only in the last few years that we stumbled on this. But I think the first published connection to this was in 2006 in a Trends in Genetics paper, where they used a computational approach to look for these gag-like genes. And, of course, there was no real reason for neuroscientists to be paying attention to that. But now that we knew, when we did know to look, there had been some work on this.
So is that what then got you full on into this protein, or had you already been focused on it?
No, I was always interested. I mean, so in grad school, I worked on trying to figure out what the protein was doing in neurons, and most of that was cell biology and some biochemistry. And then in my postdoc, I moved on to looking in vivo and trying to figure out what the protein was doing in the brain in vivo. So the logical thing to do in my own lab was to continue figuring out what it's doing. But how we stumbled on the capsid stuff is sort of a interesting story. So we weren't looking for this at all, and kind of like telling the story for students because it basically stemmed from an experiment that we thought failed, or that we were just bad at doing. So what we tried to do was really do some biochemistry where we could purify the protein. And at the time, Adam Frost's group was at Utah, and he came from Peter DiCamilli's lab, who sort of studied these endocytic proteins, dynamin and endophilin. And during grad school, we identified that ARC could interact with these endocytosis proteins to regulate synaptic proteins at the synapse. So the first idea was just, we'll purify ARC, we'll purify these other endocytic proteins and just see how they interact in vitro. So we started doing that, and my technician, Nate Yoder, started these experiments with Adam, and decided to just use affinity purification, tag the protein, make a lot of it in bacteria, pull it out of bacteria, and use straightforward ways to purify. So we did that, and everything looked good. We can get it to express in bacteria. And then the last stage that you usually do to clean up the prep is to put it through a column that can sort out things through size, so a size exclusion column. And that was when we started to get the interesting data. So we, of course, knew what size the monomer should be of ARC. And in our first experiments, none of the protein was coming out as a monomer. It all came out in the void fraction. So that meant it was so big that it wasn't even getting through the column. And so we thought, okay, well, this is just a sticky protein. Tried a whole bunch of different buffer conditions, everything we could, and none of those conditions really led to a pure monomer, and so that's where we also thought that maybe we're just not doing this properly. So then Nate, so since Adam was an expert in electron microscopy, Nate thought, well, why don't I learn how to do EM through, with Adam, and let's just take the protein and look at it, and it's probably just aggregating, because that's the most likely explanation, that it's looks like that it's so big. And so he did that, and literally the first images we got off the EM, we see these soccer balls, and we're like, what the hell are these things? And so that was the turning point. I mean, I think there's a few times in your career where you like get some data and you completely change how you see something. And I knew when we saw those images that either this was an amazing discovery, that this is completely different to what we thought we would see, or it was an artifact. And actually initially thought maybe the bacteria were infected with a virus, because even at the time we're sort of like, well, they kind of look like viral capsids, but they can't be. Why would this neuronal protein?
So you were thinking viruses right away when you saw them, right?
I wouldn't say right away. I mean, I think actually because of the endocytosis idea, we thought maybe ARC was a new kind of clathrin coat. So that so structure on the outside of membranes, not the inside. And but Adam was in the biochemistry department, and of course we have great virologists in that department, like Wes Sundquist and Chris Hill, and they ended up seeing some of these images, and in the end they're like, they kind of look like HIV. You may want to look into that. So that's what started it all. It was sort of a completely different goal. In fact, we haven't even got back to the whole endocytosis interactions. Adam got poached by UCSF and he left, and so then we ended up just pursuing this on our own. But he was instrumental in sort of launching this and taking us in directions that we would never expected. So
that's actually a figure in the paper, the electron micrographs, right? And you also have the Drosophila arc. So was that already known? That wasn't known to me.
No,
particles,
so
right?
yeah, that was another piece of luck. So no one had really worked on the fly homologue for a long time. And so I was giving a talk at this Gordon conference, and in the same session, Vivian Budnick was giving a talk, and she went first, and she started talking about the fly arcs, and I'm like, what is going on? And as she's getting more and more into her talk, I'm just sitting there going, this is insane, because she's describing biology that I thought we were too crazy to pursue. And so basically they had discovered that, so they had been working on exosomes, which are these extracellular vesicles, these membrane-bound packets of protein and RNA that get released from cells in flies, in neurons. And they took an unbiased approach to see which are the most abundant fly proteins in these vesicles. And that's how they actually pulled out the fly arc protein and RNA as being most abundant, one of the most abundant proteins in there. So they had sort of stumbled on a very different way. But they had no idea about the capsids. So we had this. So they had identified what with a released protein, and we had identified the fact that it was making a capsid, and so then put together it was made sense that the fly protein was also probably making a capsid, and so after that meeting, we kept in touch, and basically both our science, was informed by each other. We went back to look at the fly protein, and indeed it does make a capsid. And they went back to see if they could observe these capsid structures in the fly. And I think that's how it's great to see that. I mean, that's how science should work, where you sort of share unpublished data.
Sure.
And, instead of competing with each other, you end up working together. And so we actually co-submitted our papers together to Cell, and that's how they came out together.
That's right. The other article, the fly paper is in here as well.
Right.
Yep, same issue.
Right.
Yep.
Exactly. But yeah, but evolutionary speaking, it does seem like the fly, and this is also mind-blowing to me, the fly gene, the only real homology to the mammalian gene is this capsid domain, this gag-like domain. But there's no equivalent in other insect lineages that we've been able to determine. So this is a much more recent repurposing or adaptation in the fly lineage. But the chances of us, Drosophila being a model organism, it's still mind-blowing to me that our model organisms have this gene.
Yeah.
Yeah.
So tell us a little bit about the protein. So the arc, the mammalian arc, is part of GAG, right? It's not the whole thing, right? It's got capsid, which is the structural component of the viral capsid, if this were a virus.
Right.
Although these are actually ancient. These are before retroviruses evolved. So these are precursors to retroviruses. And then there's a matrix.
There's
domain,
maybe a matrix-like domain in the N-terminus, exactly. So
it's capsid and matrix. There's no NP or anything else.
There's no NC. There's
NC,
no pol.
Yeah. Right.
There's no bits and pieces like the LTRs from the original retrotransposon.
And the fly has a little bit more C-terminal, right?
The fly basically, so it's smaller and it's just the CA domain, really. There's no MA. Mm-hmm. So the N-terminus is much smaller.
And no NC either in the fly?
NC. Okay. Well, so DOC1, and this is actually in this paper that we just published today, DOC1 has a zinc finger domain that is similar to NC, but DOC2, so there's a duplication in fly. DOC2 actually doesn't have that zinc finger domain, and you can, from the structures, you can determine that there's differences in the inside of the capsid.
Okay. So from your phylogenetic analysis, you can tell that the mammalian arc is related to an old transposable element, which predates retroviruses, right?
Exactly. So that's
TY3, right?
TY3. Yep. The gypsy TY3, which is, I guess, originally an ancient yeast transposable element.
So these are DNAs that encode RT and jump around the genome by that virtue, right?
Right.
And they also happen to encode GAG. Why they need to make particles is not clear, but
yeah, exactly.
It's all there.
I think that's fascinating too, because I guess when it comes to the functional differences between retrotransposons and retroviruses, it's somewhat semantic in that these retrotransposons can form particles, these capsids. The difference is they don't get out of the cell usually, so they can't infect other
I think
cells.
Envelope is the defining feature of a retrovirus, right? That allows it to infect another cell, having an envelope glycoprotein in the membrane. But GAG by itself, it can form particles with envelopes. But your particles are different. They don't have envelopes, right? They're naked.
Well, they do. So that's where the extracellular vesicle comes in. So we do think that when the capsid gets out of a neuron, it is enveloped, does have a membrane around it. And this is part of our current research, is to try and figure out where do the capsids form, how do they actually get out of the cell, what kind of membrane are they recruiting. And so we think then, of course, that whatever is on the membrane is probably going to dictate where the specificity of uptake as well. And I think one of the more, bizarre results from our original paper was that if you purify the naked capsids, they're still able to get inside the cell into neurons, even without a lipid membrane. So we don't exactly know how that works. It's endocytosis dependent. There could be a receptor on the neuron that interacts with the capsid in some way. But
is there a difference in the efficiency of uptake with a membrane or without?
Yeah, no, that's a good question. I mean, we think with the naked capsids, the experiments we've done so far, we can take purified capsids, or we can take those extracellular vesicles and then concentrate them. Both seem to be able to transfer OC's own mRNA into naive cells. The efficiency, we haven't really got a good quantitative handle, because the problem with the extracellular vesicles is that only a subset have ARC protein in them, and so we haven't been able to purify the vesicles that only have ARC in them. But what was interesting is that the dynamics seem similar. So we could see uptake of the protein within an hour, but the RNA was only observable a few hours later, and we think that's probably because the capsid has to disassemble for us to get access to the RNA. And that seems to be happening in both the endogenous vesicles as well as the purified capsid.
So where do you think the vesicles are coming out? The dendrites, the axons, or both? And do you think then the receptors are in the postsynaptic regions? Or?
Yeah, so ARC's only expressed postsynaptically, at least to high levels. And one of the actually interesting things another reason why ARC was interesting is that a lot of its mRNA is trafficked out into those dendrites, and it's then locally translated at synapses or near synapses. And we've known for a long time that many RNAs get trafficked out from the cell body of neurons, and it makes sense because their processes are usually quite far away from the cell body. So this is another mechanism to really make protein at synapses very quickly. So ARC was one of the most robust mRNAs, or the highly expressed RNAs that get out into the dendrite. So our model right now is that we think that when you get local translation, there's enough protein there to form a capsid, and that it would get out at the dendrite. Okay. But once it's out of the dendrite, then all bets are off, and we're not exactly sure where they go in the intact brain.
Can you see these in mouse brain sections?
Yeah, we've looked. So we could see things that look like capsids, but we haven't, I would say, definitively shown that it's ARC. And so that's what we're working on now, is being able to tag the protein in a way that we could do light microscopy and then an EM to really say, okay, this is where we see the protein. We see capsid-like structures. One of the problems is that we think...
And your control could be the knockout mouse.
Right, exactly. But, unlike a virus, which obviously infects multiple copies into an infected cell, here we think that you need about 150 monomers to make one capsid. So you need a fair amount of protein. So we don't think at any given time there's going to be a ton of these things being made, which in itself is, I guess, interesting in terms of what its function could be.
So the transcription of ARC and the translation is regulated by neuronal activity. Do you think the release that is also regulated by activity? Yeah,
so we have evidence for that. So if we increase activity, we see in neurons, we see more ARC being released. Okay. So we do think that part's consistent with it being activity dependent.
If I remember, you have a version of the protein which doesn't form particles. Is that right?
We think we've just stumbled on we think we've made a point mutant that will block assembly.
Okay.
And so we're hoping that's going to be the tool to sort of tease out these non-cell autonomous versus autonomous.
Yeah, you should put that back into knockout mice and see.
Exactly.
If the memory is fine, then maybe you don't need particles.
Right, exactly. So the two models here, or the two big models, are one that it is involved in memory. I would say from the memory field perspective, if that is the case, we're going to have to rethink of how memory works, because for most models rely on the fact that you have synapse specificity, not even cell specificity, but synapse specificity for which circuits are made. And so this is actually why I was somewhat resistant to the idea that ARC could be released from cells, because we knew so much about how ARC protein was regulated, in that it was only made at certain synapses and it would only traffic to certain synapses. But then once it gets out of the cell, you lose that synapse specificity. So,
although if it's such few capsids and it's made at the dendrites that got activated, like locally translated, then if you have so few capsids, then they can't go that far, right? They would only reach the presynaptic partner, maybe.
Right.
Just by the nature of there being so few of them.
That could exactly. So it could be that it's a retrograde signal from the post to pre. Yeah. And there are models of memory that sort of include that kind of signaling. We just don't know yet.
Right.
But the alternative model, of course, is that this intercellular signaling has nothing to do with memory? And if it's not, then what is it involved in? And that's going to be an interesting question to address. One interesting tidbit of data is that we see ARC vesicles or ARC in extracellular vesicles in blood.
Mm-hmm.
But we don't know yet if that's the source. So we don't know if they're coming out of the brain or are they being made in a peripheral cell or,
Well, it's clear that the gene is involved in memory, right?
It's clear that the gene is involved, yeah.
Because the knockout mice have these deficits, as you said. But
exactly.
Whether you need to make a capsid-like structure where it's just a protein, that's really and that's why that mutant would be really interesting.
Yeah,
to look
exactly.
at, yeah.
So we're definitely excited about having a fairly clean mutation that would only hopefully disrupt one aspect of the protein.
So you also feel that these capsids contain RNA, right? So how do you show that?
Yeah, so when we purify the capsids even from bacteria, we could detect RNA.
Mm-hmm.
If you just even do an RNA, if you look at the protein to RNA content in the capsids. So when we try to get at the RNA, we predicted that maybe ARC mRNA would be a logical candidate. And so we used qPCR to show that indeed, even in the purified capsids in bacteria, they are encapsulating ARC mRNA.
Mm-hmm.
And then we looked at extracellular vesicles from neurons from rat, and that was the same case that we could detect ARC mRNA that was protected, we think, inside the vesicle. And so then the final experiment was to show that if ARC is actually encapsulating its own RNA, could it transfer it into knockout neurons? And that is also the case. So even the naked capsids that have ARC mRNA from the bacteria are able to transfer that into these ARC knockout neurons. But we did detect in bacteria some ARC capsids that had bacterial RNA. And this is actually similar to HIV gag in that there is some specificity for interacting with its own viral RNA. But if you take out the viral RNA, it'll encapsulate host cell RNA too. So we're still sort of trying to figure out what the specificity is here. But we also think that there's probably other RNA species inside the capsid.
Yeah, because the viral RNA has a packaging signal that makes it more efficiently encapsulated. Because
exactly.
If it weren't competing very well with cellular RNAs, it would be a lousy virus.
That's right. That's right.
But I wonder if the ARC mRNA itself has some remnant of a similar sequence, right? Yeah.
So that's what we're trying to figure out now. The fly ARC does seem to have some sort of signaling in its 3' UTR, but for the rat, we're not sure yet. But yeah, so one of the big questions we're trying to address now is what is the actual endogenous cargo? Is it just ARC mRNA, or are there? We have hints that it's probably not just ARC mRNA; that there's probably smaller non-coding RNA species inside. But.
And do you think both ARC and those are involved in the memory effect? Is that what you're thinking?
Yeah. Well, that's what we think. But how is the next? I mean, it's once we've identified it, we think what's inside the capsids. I think we'll get to that. But if the ARC mRNA is the main cargo, there are some hand waving explanations for why you'd want to do that.
Yeah.
But yeah.
Sorry, go ahead.
From the crystal structure, can you estimate how many molecules it can pack of RNA, or how long and how many each capsid? Yeah.
So. John Briggs in Cambridge has reconstructed the fly ARC with cryo-EM, and I should know this, but off the top of my head, I don't know what the capacity is.
So it's about 30 nanometers, it looks.
Yeah.
Right? So that's polio, which is sitting here on the table between us. This is a 30 nanometer capsid, and it can hold a single RNA 7,400 bases long.
There you go. Okay, so
that's pretty maximally packed into there. So that's about it. We could put smaller ones in too, of course. Well, and the ARC mRNA is probably much smaller than that.
Yeah, so the ARC mRNA, the fly ARC protein's only 200 amino acids.
Yeah.
The rat ARC capsids, I think, are a little bigger, so they're about 50 nanometers in average, 50 to 60.
Okay.
So they could probably package 10 kb maybe.
So you actually don't know. So there's this interesting memory effect that if you take out the whole gene, the mice have a problem. But in terms of what parts of the protein are involved, you don't know, right?
That's right.
That would be interesting to know. Of course, there are parts of the protein that are involved with particle formation, and as we said, you can sort that out. But beyond that, if the ARC itself is involved, then certain parts of it should have different activities, and I'm sure you want to look at that. But that's hard, right, to do that?
Well, actually, we know a lot about that. So that's
okay.
Okay. If anything, we know way more about that. So that actually was my PhD thesis, and so what we found was that ARC protein can interact with these endocytosis proteins, dynamin and endophyllin, to regulate glutamate receptors at the synapse. And so if you overexpress ARC artificially in neurons, you downregulate synaptic function through trafficking of these glutamate receptors. And all of this actually made sense in terms of models of memory, because we think that the basic way of memory encoding, as we talked about in an earlier episode, is through the synaptic plasticity. So it made sense that ARC itself, if it had a memory function, was doing it through synaptic function. The only issue to me, though, is that in terms of explaining the persistence of memory, this is where, again, is a bit of a black box, because during encoding, when you're learning, it makes sense that these synapses are changing right away. They form new circuits, and those are the circuits that really mediate the memory. But why do you need hours of consolidation? So this process of consolidation, molecular consolidation, is slow. And you need protein synthesis, so you need new gene expression. And the idea would be that whatever those consolidation is doing is it's stabilizing the strength of these synapses. But how that actually works is not super clear. So this is where I think we're in unexplored territory with the cell-to-cell signaling, and whether that second function of the protein. So we think that synaptic trafficking role is probably done through the monomer. And in fact, we have some data where some of the protein interactions at the synapse will block the ability of ARC to form an oligomer. So there's probably switches in function that tell it to stay a monomer or become a capsid. Although in bacteria, the default seems to be that it'll spontaneously make these capsids. So that seems to be like it wants to do that.
One of the experiments in this paper, well, first of all, what's interesting is you show that the RNA is actually needed for assembly of the capsid, right?
Right.
Which is funny, because usually we think of just producing GAG and you get capsids that are empty, right? So what are your thoughts there?
Actually, I mean, HIV GAG, to get a proper formed capsid, you do need RNA interactions. Yeah.
Viral RNA. And it's thought that some of this is that the viral RNA seeds the capsid assembly in some way. So that's actually, so the biochemistry there is quite similar to how ARC is potentially. Because not all retroviruses are like that, right? Some of them you can make. In fact, retroviral vectors work because you can make empties and then put some other gene in them, right, when you're ready. So that's interesting. And then there's an experiment which maybe you can explain, because I don't understand the biology behind it. The transferred ARC can undergo activity-dependent translation. What does that mean?
Yeah, so that was our attempt to say, okay, if we can see RNA getting into the cell, is it accessible to be translated? And this goes back to, we know that, especially in cultured cells, which are really active, there's a lot of ARC mRNA in those dendrites. And we know a little bit about the signaling cascade, which receptors you need to activate to get translation of that RNA in the dendrite. So what we did with the transferred RNA was say, well, does the same drug that would induce endogenous translation induce translation of the transferred RNA? And that's where we did see an increase in protein when we add the drug, even in the knockout, suggesting that the transferred RNA was accessible to be translated.
I got it. Okay. Very good. So here in your discussion, this is why I mentioned before that they're not, you say in the absence of an envelope, this transfer occurs. But you said they are enveloped. So are they? Yeah.
So that's where so with endogenous vesicles, the way we purified the vesicles was...
Oh, I see. You have a problem in distinguishing them from the EVs.
Right, exactly. So,
okay.
Yeah, this is where, the field of exosomes has sort of become murky because, even in as you probably know more than I do, some of these so-called naked viruses actually hijack exosomes.
Sure.
And so they become enveloped.
Yeah, we have learned now that, hepatitis A virus, which was considered classically a naked, very much like poliopocornavirus, in certain conditions can acquire an envelope. Yeah. And, of course, when it's in the bile duct and the intestine, it doesn't have the envelope because it's digested by bile salts, right? It's dissolved. But it's thought that transmission occurs in these pseudo-enveloped, quasi-enveloped particles, right?
Right.
And there's evidence that even polio and some other naked, but they're not actually enveloped because, viruses that have an envelope, a functional envelope, it has glycoproteins in it so that it can attach to host cells. Because without that, there's no way to ensure specificity.
Right.
So something else is going on in the case where there's a quasi-envelope. But yes. So you're thinking that it might be envelope, but you're not sure yet. I think earlier you said you're looking at that, right?
Yeah, no, I think we have solid evidence that ARC has an envelope around it, or it has a lipid membrane around it.
All right, because even with Hep A quasi-envelope, it gets into cells by mechanisms that are being determined. Stan Lemon is at UNC Chapel Hill, and he's a person trying to figure that out, and you might get some clues from him.
Yeah,
what's happening here, because maybe there are proteins in the ARC envelope pulls from the cell, and those are helping it get into another cell.
Yeah, exactly. And I think it'll be interesting. So, because there's another paradox or puzzle here, which is how does ARC get the RNA out of the endosome once it gets taken up? So, viruses use fusogenic proteins on the envelope, but what's the equivalent here? We don't know.
So you're pretty sure that these get into endosomes, right?
Right.
Yeah. So they're in you think that the actual ARC particle is in the endosome in some way, right? And it's probably naked in there, right?
Right.
Because this envelope would bring it in, but then it has to get out, yeah. Could have a different mechanism. I mean, poliovirus is naked to begin with, and it's taken into endosomes. And then the particle actually forms a pore in the membrane.
Right.
And the RNA comes out through that.
Right.
And that pore is driven by the receptor interaction. So this happens very soon after the particles get endocytosed.
Right.
But the receptor binding to the particle, which it's a model of it right there. That's polio with its receptor. Huh. If you just add receptor to naked viruses, all the RNA pops out because this channel forms. And if this is in an endosome, then the RNA goes through a channel into the cytosol. And so similar things could be happening.
Yeah. No, I think that's, I mean, our model is potentially that could, how ARC is being disassembled, how it gets out of the endosome. These are all sort of, for neurons, this is unexplored territory because you're talking about things that are usually done with viruses. So
it's interesting. You can learn a lot from viral studies, right? But this probably is a mechanism that predates viruses.
Right.
Although let's say it's ancient. I don't know which came first. I suspect viruses often steal things, although they don't steal. They are a lot of their mechanisms originated in cells, right? So not sure they invented anything in the end. But
Well, I think from being a non-virologist, the fascinating thing to me here is that that usually you think of viruses as being bad, and they of course are bad to hosts, but on the long run, they've provided material for evolution to use, and he has something as complicated as memory that may have arose from, or at least this kind of complicated memory arose from an ancient retrotransposon, and that's crazy.
yeah, so that would be previral, but the other example is syncytin, which you mentioned, which is an envelope like a protein that's been exapted to form the placenta. That is certainly a viral derivation.
Right.
And you probably know this, Andres, these viral fusion proteins are related to the gamete fusion proteins.
Yeah.
Right. In like Tetrahymena and other organisms like Chlamydomonas. The fusion mechanism is similar.
Yeah,
it's really cool.
Yeah, it's amazing that there's rules that evolution follows. I don't really know what those rules are, but there's constraints, right? So there's the syncytins they've been adapted or repurposed at least 6 times or something, right?
Yes, absolutely. It's not just once.
Yeah,
in different lineages.
But always for the placenta.
Yeah,
so it's like the only best way to get the placenta.
Well, you need to fuse the syncytial trophoblast, right? That outer layer is all fused, and you use a fusion protein to do that, and this works really well. It's very cool. There are other examples of these either pre-retroviral or retroviral proteins being exapted for cellular functions, and it looks like this is an ARCs or another one.
Yeah.
So in that sense, the two ARCs, the Drosophila and the mammal, as you point out, they seem to have different transposon ancestors, right?
Right.
So this happened at least twice.
Yep, at least twice. So, and we think that some of these other gag-like genes may also still maintain the ability to form capsids.
Mm-hmm.
And if that's the case, then you'd think maybe there's a repertoire of different capsids that get expressed at different cells, carrying different cargo. And this is that ARC is not unique in the way that it's signaling.
Right.
So we're excited to sort of pursue that.
So if the ARC capsids or envelope ones have a receptor, how will you go about finding the receptor?
Mm.
Yeah. Well, so one way we thought about is, so it looks like uptake and transfer efficiency of all of this is very good in neurons, but not in non-neuronal cells. So if you take these capsids and just dump them on cell lines like Higgs cells or HeLa cells, they're not very efficient at taking them up. So you could have a gain of function screen to look for things that we could add back to those cells, or we could try to do old school virology, I guess, which is when they were looking for receptors for these capsids. So I'm not sure if we can. My lab's going to go down that road, but maybe one day we'll have to do that.
It's tricky because if the particles get into all cells, then you have a high background to deal with. Right? So we identified the polio receptor because we had mouse cells that completely lacked it.
Right.
We could just put human DNA into them and get production of it and select it that way. But it's going to be a little tricky if it's in all cells or it can bind to all cells. Maybe the specificity is low. That'll be tough. If you can find one day a cell line that doesn't bind and take them up,
right,
then you could use that.
Yeah. No, I think even the Higgs cells, they're really not very good at taking them up. Certainly the naked capsids. So I think we actually could find cell lines where we could do these screens to add back things. Is
there any species specificity? So will human ARC particles get into mouse cells?
Yeah, so that's a good question. I don't think we haven't tried that experiment to make human. The conservation, even at the amino acid level, is really high. It's like 90%. So my prediction is that the naked capsids are probably going to get into the mouse.
Yeah.
Because these things have not been as refined as the viruses for species.
Now you could try very distant ARCs in cells. You could even human ARC into fly cells. Maybe it won't bind or something, or
Yeah,
vice versa.
I think we could definitely try. Certainly, that we could purify the fly capsids to see if they could get into the rat cells.
So fly capsids get into fly cells, but not human cells. Then maybe you have a handle to do that. So that would be interesting. So the authors on this paper are interesting. So Cedric Fischott, he used to be at Utah, right?
Right.
He's been. We've done some of his papers on TWiN. So he's interested in this whole exaptation of viral genes, right?
So Cedric's group. So Cedric was at Utah when we were sort of stumbling on this biology, and so I recruited him because he is this expert on these repurposing events, these transposable elements. So it's his group that did the phylogenetic analysis in that paper.
Okay.
Yeah.
And then there's some people. So he's at Cornell. Some people in Denmark, or is that someone who just went there afterwards?
Yeah, so that's Simon. So the other group that we collaborated with is John Briggs at the MRC lab, who helped us with some of the EM.
Got it.
And so it's his group that just published this cryo-EM structure of the fly arc in Nature Neuroscience.
Oh, look who's here, David Belknap.
Yeah.
He's at Utah, right?
Uh-huh.
I think I met him when I went out there. So that structure, he was a collaborator. He used to be with Alistair Stephen. He was a cryo-EM structure person at NIH. So he helped us do that. So what did he do here?
Yeah, so David runs our EM core.
So
he was instrumental in helping us get that up and running.
So then you have, just this morning, the structure paper in Nature Neuroscience, structures of virus-like capsids formed by the Drosophila arc proteins. Now, why did you do Drosophila and not the mouse?
So the simple question there is because we couldn't get the mouse to work.
You couldn't get enough particles, or just...
No, so it turns out that for whatever reason, the mouse capsids are much less homogeneous in size.
I see.
Which I gather is actually similar to HIV gag. And so we turned to collaborating with John Briggs because they had been able to use sophisticated tomography to get at the HIV gag structure. And so that work's still ongoing. So the original goal was to look at the mammalian arc, but in the meantime, turned out that the fly arc could make really homogeneous. So once you've got the preparation homogeneous, it took them like a couple of weeks to get a structure.
Yeah.
This is the amazing thing about cryo-EM. So the simple answer there is that it worked for the fly, and they were able to get up to 2.8 angstrom resolution, so really high resolution, almost as good as crystal structure, but for the whole particle. So we're still hammering away at the mammalian arc, but it does seem like the mammalian arc capsid's probably forming is different to the fly arc.
All right, so probably eventually you'll get that structure as well. The reason I ask is because these Drosophila particles have spikes.
Yeah.
It looks like a virus capsid.
It's crazy.
When I first looked at it, I said, what's going on? There's no membrane here, but I realized it's an integral part of the protein.
Yeah.
A spike, forgot which axis it's at here. Here, short spikes, 5 to 8 nanometers, protrude from the center of each capsimer, which is a structural subunit. And so these look like virus particles. I wonder if those spikes are attachment.
Yeah. No, when we saw that, we don't know if the mammalian arc has those spikes, but John, who's a virologist, right, or has been working on these capsids for these viral capsids for so long, also said if you didn't know that these proteins were from an animal, they would just say this is a new virus. It's crazy.
Yeah, sure. What's interesting is that I went and looked at the gypsy gag structure particles.
Right.
There's no spikes.
No spikes.
Which says that either this arose randomly and it had spikes and it became exapted for this function, or it developed it over many years of evolution. I mean,
yeah.
Right?
Yeah. But as you say, it's clearly it could be a site of interaction either through protein or membrane. So that's some follow-up work that we're doing. Yeah. We're actually collaborating with a group at Utah and Sophie Caron's group to look at the function of the fly genes in memory, so in the CNS. So Vivian's group had sort of looked at this neuromuscular junction synapse, which is muscle, nerve to muscle, but we want to know if the fly arcs are involved in memory at all. Because if they are, then it's another example of like the syncytins, where there's clearly a reason why you would want to have this kind of biology. But as we were talking about Andreas, well, it's not like fish can't learn. They have memory. So the big question there is, are there other proteins that are gag-like that do similar functions to ARC, or there's just alternative ways to get to the same kind of information storage?
Can the fly ARC gene complement the mouse knockout?
Yeah, so we haven't done that question. I actually think that's probably... I think that would be informative. My hunch is that the fly arc may not be involved in memory at all, but it's expressed in the fly brain. But that N-terminus of the mammalian arc is what that the monomer of the mammalian arc is where it interacts with these synaptic proteins, and that's lacking in the fly.
I see.
What about the different types of the brain, the necessity of ARC? Like when you were looking for ARC after neuronal activity, some parts express it at higher levels than others. Is the involvement in memory, you think, also different? Or
yeah, so one of the striking patterns that you see is that ARC is induced in the hippocampus of the brain, which is where we know encoding of memory is happening. But you also see it in the cortex and sensory cortex. So another part of my lab has been looking at this experience-dependent changes in sensory cortex, so visual cortex, and very similar molecular mechanisms that are used for episodic memories, the memories that we think about, are also involved in wiring up the brain in response to experience. So the classic example is that if you have issues with your eyesight, so let's say you have a cataract or strabismus as a child, if you don't correct that problem with the eye early in development and you come back as an adult, even if you correct the eye, the brain is unable to make sense of the information because that plasticity is gone. So we showed that in mice, at least, if you take adult mice and boost ARC expression, then you can actually get the brain to rewire itself in response to experience. So
I noticed that in this paper you did both dark 1 and 2, and they have similar capsids. And it says here that dark 2 has no function, no known function, right?
No known function.
But we don't know if it's involved in plasticity of any sort. Whereas dark 1, there is evidence, right?
Right. So Vivian's group only looked at DARC1.
I see.
But it's clear that, as you can see from those structures, they're very similar. There's just slight differences. So we don't know. So what we've done is we've CRISPR'd the whole locus, so we have a fly line that lacks both proteins. And so then, we can add back one or the other to see if any phenotypes are specific.
Questions that remain to be answered include whether the N-terminal spikes mediate membrane or RNA binding, and whether the zinc fingers and basic capsid interior determine RNA packaging. So on the inside, there's this zinc finger,
right,
which you think is involved in RNA binding, right? Which you could easily address, right?
Yep, exactly.
Cool. All right. Anything else we missed we should mention that's of interest?
No, I don't think so. Yeah, I mean, for us, it's been a real fun, journey, because I'm not a virologist and had no idea about any of this viral stuff until now. So, are
the virologists interested in this? I mean, I am. I'm just wondering what you've encountered in general.
When the paper came out, there was a lot of interest. It was one of those papers where I didn't really know what the reaction would be, because from the neuroscience field, it was sort of really out of the blue. from the virology kind of viewpoint, there was like, well, this is a bit bizarre. So I gave a talk at the Cold Spring Harbor retrovirology meeting in 2018. And, the only neuroscientist there pretty much. And I think there was a lot of interest, partly because I think, the HIV field has become saturated. So much about this one kind of virus. But there was, I think, a lot of pushback and skepticism because a lot of what we were showing for this, for ARC was not exactly how retroviruses work. So, for example, the non-enveloped nature of it, that sort of thing, although I think it's just because we just don't know enough about how all of this works. But there's been a lot of interest from virologists, and I think that's been really fun. I've been given opportunities to give talks to virology departments or people that are interested in exosomes and extracellular vesicles, so not just neuroscience.
Of course, you have Wes Sundquist there, who's a retrovirology structural person, right? So he must be interested, right?
Yeah. No, Wes has been great. In fact, Wes's expertise were there's no way I could have navigated the HIV field without his help and his labs. In fact, my lab is situated below the medical school, and we don't have any ultracentrifuges either in my floor, and so we have to go up to Wes's lab to use his equipment. But he's been super generous. This is one of the things about Utah that's been great is, one, I was lucky to have these kinds of scientists that had the right expertise, but two, that they're open to collaborating and helping. And so again, I think a lot of things had to happen for this story to sort of, evolve itself. And one thing was being at a place that's very collaborative.
Yeah. So your host here was who? Who invited you here?
Eric Kandel's group.
Cool. Have you spoken with him in the group at all yet?
Tomorrow, yeah.
Tomorrow?
Yeah. I'm excited to meet him and talk to him.
He's the memory guy, right?
That's right.
So I wonder what he thinks of viruses in memory or virus-like particles.
Well, he's, he had this really cool idea of memory persistence a few years ago about prions. And I don't know if you saw this, but one idea at the molecular level to sort of sustain activity was that if you had a prion-like mechanism that would be self-replicating. And he showed that there was something like this in a protein called CPEB that can affect translation of these dendritic RNAs. And so I think Eric's a fan of these out-of-the-box ideas, but we'll see tomorrow, I guess.
I wonder if there are ARC particles in aplesia. Probably not from what you said about the fish, right? Yeah,
so that's where I think that there's no ARC gene in aplesia, but I don't know if there's any gag-like genes, though. We haven't looked for sure.
Well, most organisms have these transposons, retrotransposons, right?
Yeah, and in fact, these endogenous retroviruses, which is another topic that I'm sure is interesting in that our own genomes are riddled with these viral remnants, and some of them get reactivated during disease, or there's some correlation with them during disease that is emerging, especially in neurodegeneration, for example.
Yeah, they're there. They're in mollusks.
Oh, yeah?
Yeah.
See?
No. You never know. All right. We have just 2 emails here I want to read before we wrap up, both from Anthony. And the first one has to do with this quote from Mendel that we read last time. Mein Zeit Wort schon kommen. He writes, Mendel himself, though grievously disappointed at the neglect of his views, never lost confidence in them, and often exclaimed to his friend that German phrase, which I forgot what it means. Let me search it. Mine. Here we go. I'm going to translate it because I don't know what it means. I think we did that last time. Let's see. Google Translate. We'll put it in here. My time will come.
There you go.
Very good. My memory is reading that was Mendel's comment after receiving a withering rejection letter from a journal. Yes, my time will come. I, of course, meant the reference as hyperbole and humor. And then Anthony also writes, dopamine risk-taking and toxoplasmosis. Humans with latent toxo display altered reward modulation of cognitive control. This is a paper from Scientific Reports in 2017. Latent infection in humans, and the quote is, taken together, individuals with latent toxo show superior behavioral performance in challenging cognitive control situations, but may at the same time have a reduced sensitivity towards motivational effects of rewards, which might be explained by the presumed increase in dopamine. So toxoplasmosis somehow is fooling with that.
We need more episodes on infections and the brain and neuroscience.
Yeah.
Viruses, parasites.
It's hard to know. I mean,
well, those are the parasites that come from cats, right? And
Yes.
there's actually some evidence that cat owners are predisposed to schizophrenia.
yeah,
But
it's
this
pretty
is
tenuous.
a really weak link.
Yeah. The problem is a good fraction of the world's population is infected
Right.
with toxoplasma organisms, and so it's hard to tease out, especially the French who like to eat raw meat. That's a risk factor.
Yeah.
We've talked about this on our parasitism podcast, and there's a lot of skepticism there about whether, but there's no doubt that in mice it makes them immune to cat urine.
Yeah.
Smelling cat urine, so they don't run away. They don't
Right.
smell it, so they stay there, and they get eaten,
Right.
and that gives the cat the infection. So whether that's an effect of the parasite or not, who knows? Hard to know. All right, that is TWiN at microbe.tv/TWiN. You can find the show notes and links. If you have questions or comments, please send them to TWiN, TWiN, at microbe.tv. And as I mentioned earlier, if you like what we do, we have a whole bunch of podcasts here. Consider supporting us financially. Go over to microbe.tv/contribute. We can use PayPal or Patreon just to give us a little bit of money a month, $1 a month. Sure. That would be great. Or if you want to give a certain amount of money once, you could do that at PayPal through microbe.tv/contribute. We'd appreciate your support because we do have expenses, and that's what the money would go for. Today on TWiN, we have had Andres Bendesky. He on Twitter is Bendesky. Thanks, Andres.
Well, pleasure to be here.
And special guest in studio, Jason. I always have trouble with Jason. Jason Shepard. Jason Synaptic on Twitter. Good to see you in vivo.
Yeah, nice to meet you in
Thanks
person.
for coming by.
Thanks, Vincent.
You going back downtown now, or what are you doing?
Yeah, so I'm at the 79th Street from my hotel.
That's right. That's the hotel we use for everyone. It's not bad, actually. What's the name of it?
The Lucerne?
Lucerne, yeah. Lucerne. It's good. Are you going to bring him back so he doesn't get lost?
Yeah, he won't get lost.
I'll figure it out.
Well, your lab's back down there anyway.
Yeah.
That's
I'll put him on the right way.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening. Yeah, I got a neuron there for you. I meant to show you that.
Yeah.
It's one of those giant microbes, right? Yeah. I usually kill them. I said that many times here on the show. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back soon.